class. Last time we have learned about the bacteria endospores. In this lecture, we are going to learn about the structures external to cell wall. Among the possible structures external to the prokaryotic cell wall are the glycocalyx, flagellar, axial filaments, fibrillary, and pili. Many prokaryotes secrete on their surface a substance called glycocalyx. The bacterial glycocalyx is a viscous, gelatinous polymer that is external to cell wall and composed of polysaccharide, polypeptide, or both. Its chemical composition varies widely with the species. For the most part, it is made inside the cell and secreted to the cell surface. If the substance is organized and is firmly attached to the cell wall, the glycocalyx is described as a capsule. The presence of a capsule can be determined by using negative staining. Capsule staining is more difficult than other types of staining procedures because capsular materials are water-soluble and may be dislodged or removed during rigorous washing. To demonstrate the presence of a capsule, a microbiologist can mix the bacteria in a solution containing a fine-colored suspension of colored particles to provide a constructing background, and then stain the bacteria with a simple stain, such as safranin. Because of their chemical composition, capsules do not accept most biological dyes, such as safranin, and thus appear as holes surrounding each stained bacterial cell. The glycocalyx can help bacteria adhere to its environment. The glycocalyx protects bacteria from dehydration and loss of nutrients. In certain species, capsules are important in contribute to bacterial violence. Capsules often protect pathogenic bacteria from phagocytosis by cells of host. Some types of glycocalyx can be important in formation of biofilm infections. Glycocalyx also facilitates communication among cells. Many motile bacteria move by use of flagellar, thread-like locomotor appendages extending outward from plasma membrane and cell wall. Although the main function of flagellar is motility, they can have other roles. They can be involved in attachment to surfaces, and in some bacteria, they are valence factors. Bacterial flagella are slender, rigid structures about 20 nanometers across and up to 20 microns long. Flagella are so thin, they can't be observed directly with a bright field microscope, but must be stained with techniques designed to increase their thickness. A tedious and delicate staining procedure uses a mordant and the staining carbofortin to build up the diameters of the flagella until they become visible under the light microscope. Microbiologists use the number and the arrangement of flagella as a diagnostic aid. Transmission electron microscope studies have shown that the bacterial flagellum is composed of three parts. First, the longest and most obvious portion is the filament, which extends from cell surface to tip. Second, the basal body is embedded in cell envelope. Third, a short, curved segment, the hook, links the filament to its basal body and acts as a flexible coupling. As the figure shows, the filament is a hollow, rigid cylinder constructed of subunits of the protein flagellin, which ranges in molecular mass from 30,000 to 60,000 Daltons, depending on the bacterial species. The filament ends with a capping protein. Some bacteria have fins surrounding their flagella. For example, Vibrio cholerae flagella have lipopolysaccharide fins. Individual flagellin subunits are transported through the hollow filament. When the subunits reach the tip, they spontaneously aggregate under direction of a protein called the filament cap. Thus, the filament grows at its tip rather than at the base. The hulk and base body are quite different from the filaments. Slightly wider than the filaments, the hulk is made of different protein subunits. The base body is the most complex part of a flagellum. The base bodies of E. coli and most other typical gram-negative bacteria have 4 rings, L, P, MS, and C, which are connected to a central rod. The L, P, and MS rings are embedded in cell envelope, and the C ring is on the cytoplasmic side of the MS ring. Typical gram-positive bacteria have only 2 rings, an inner ring connected to the plasma membrane and an outer one probably attached to the peptidoglycan. Bacterial species often differ in their patterns of flagellar distribution, and these patterns are useful in identifying bacteria. Monotrichous bacteria have 1 flagellum. If it is located at an end, it is said to be a polar flagellum. Amphotrichous bacteria have a single flagellum at each pole. Lophotrichous bacteria have a cluster of flagella at one or both ends. Flagella are spread evenly over the whole surface of Patrichous bacteria. Each bacterial flagellum is a semi-rigid helical structure the cell by rotating from the basal body. The rotation of a flagellum is either clockwise or counterclockwise around its long axis. The movement of a bacterial flagellum results from rotation of its base body and is similar to the movement of the shaft of an electrical motor. As the flagella rotates, they form a bundle that pulls against the surrounding liquid and propel the bacteria. Flagellar rotation depends on cells' continuous generation of energy. Bacterial cells can alter the speed and direction of rotation of flagella and thus are capable of various patterns of motility. When a bacterium moves in one direction for a length of time, the movement is called a run or swim. Runs are interrupted by periodic, abrupt, random changes in direction called tumbles. Then a run resumes. Tumbles are caused by a reversal of flagellar rotation. Bacteria exhibit taxis to a variety of stimuli, including light and oxygen. The movement of cells toward chemical attractants or away from chemical repellents is the best studied type of taxis. Chemotaxis is readily observed in Petri dish cultures. If bacteria are placed in the center of a dish of semi-solid agar containing an attractant, the bacteria will exhaust the local supply of the nutrient and swim outward following the attractant gradient they have created. As Figure A shown, the result is an expanding ring of bacteria. As Figure B shown, when a disk of repellent is placed in a petri dish of semi-solid agar and the bacteria will swim away from the repellent, creating a clear roll around the disk. Fimbriae can occur at the poles of the bacterial cell or can be evenly distributed over the entire surface of the cell. Fimbriae are shorter, straighter, and thinner than flagella. They can number anywhere from a few to several hundred per cell. Fimbriae have a tendency to adhere to each other and to surfaces. As a result, they are involved in forming biofilms and other aggregations on the surfaces of liquids, glass, and rocks. Fimbriae can also help bacteria adhere to epithelial surfaces in the body. For example, the fimbriae of E. coli O157 enable this bacterium to adhere to the lining of the small intestine, where it causes a severe watery diarrhea. Pili are usually longer than fimbriae and number only 1 or 2 per cell. Pili are involved in motility and DNA transfer. Some pili are used to bring bacteria together, allowing the transfer of DNA from one cell to another, a process called conjugation. Such pili are called conjugation pili. In this process, the conjugation pillars of one bacterium called an F+ cell connected to receptors on the surface of another bacterium of its own species or a different species. The two cells make physical contact, and the DNA from the F plus cell is transferred to the other cell. The exchanged DNA can add a new function to the recipient cell, such as antibiotic resistance or the ability to digest its medium more efficiently. Okay, that's all for today's lesson.